Hello, everybody. Welcome back to the HCC Connect Science seminar series. My name is Judith Agudo. I'm an assistant professor in the Cancer Immunology and Virology department at the Dana Farber Cancer Institute. And before I start with my introduction, I just want to briefly remind everybody that the seminar will be posted at the website, at the DFHCC Connect Science website. And please submit your questions through the Q&A option at Zoom, not the chat option. And it is really my true and exciting pleasure today to introduce to all of you Dr. Stephanie Dugan. Dr. Dugan is also an assistant professor in the Cancer Immunology and Virology department at Dana-Farber Cancer Institute, and an assistant professor in the immunology department at Harvard Medical School. Dr. Dugan is, in my opinion, one of the most remarkable cancer immunologists I have ever met. She has been working in cancer immunology for a very long time, starting from her PhD here in the University of Harvard with Glenn Dranoff. Before cancer immunology was this major breakthrough, she was already working in cancer immunotherapy. After obtaining her PhD from Harvard University, she went for her postdoctoral training at MIT with Dr. Hidi Plu, working in a really outstanding technique where she developed this tool that allowed her to literally clone mice out of B cells or T cells to develop these BCR, B cell receptor, or TCR, T cell receptor mice. And as an independent investigator, she has been highly prolific with many papers in cancer immunotherapy, which has granted her multiple very prestigious awards, such as the Pew Scholar Award, the Smith Family Award, and the Pancreatic Cancer Action Network AACR Pathway to Leadership Award. In her lab, she works in various aspects related to cancer immunotherapy with a major focus on pancreatic cancer, which is something that I truly admire, because while many of us work in tumors in which immune responses we know already happen, she's committed to find a cure for this devastating disease in which immune responses are so hard to get. So she went for the hard one. And not only is she a highly prolific investigator, she's also a really remarkable, outstanding mentor, which granted her also this Harvard Medical School Mentor Award, Mentor Award of the Year. And I can tell you, if anyone in the audience is looking for an outstanding mentor for your postdoc, don't doubt, reach to Dr. Dugan. She's really committed to mentor and train the next generation of cancer immunologists. And without further ado, please, Dr. Dugan, I'm looking forward to your seminar.
Oh, well, thank you, Judith, for that very touching introduction. So I am a cancer immunologist, but do like to start my talks reminding people that the primary function of the immune system is to defend the body against pathogens. And so this is what it evolved to do, and anything else we can convince it to do with therapy is really a side effect of this primary function. So T cells can also recognize cancer. And just to put everybody on the same page, they do this through antigen-specific receptors that can recognize specific peptides, either from a virus or from the tumor, that are presented on MHC class 1. MHC class 1 consists of a heavy chain that actually binds the peptide via these anchor residues, and a light chain beta-2 microglobulin. And so we know now, in the age of cancer immunotherapy, that we do have means of reactivating T cells to recognize and kill tumor cells. These are conventionally called checkpoint blockade. And I just want to go through a diagram of how our current understanding of how checkpoint blockade works. And so we start with a tumor that usually at baseline doesn't have very much MHC class 1 on its surface. But as some of these tumor cells die, they can be picked up by dendritic cells that can prime naive CD8 T cells that are tumor specific and can make a little bit of interferon gamma. Perhaps they're recognizing a neoantigen or some other antigen expressed by these tumors. The interferon gamma induces upregulation of interferon gamma response genes, notably MHC itself, as well as PD-L1. And so at this point, T cells still don't kill these tumors because they're blocked by the expression of PD-L1. But of course, we have antibodies to PD-1 and PD-L1, and we can therapeutically allow CD8 T cells to come in, recognize MHC class 1 on the tumor cells, and kill them either by direct cytolysis with perforin granzyme or by production of interferon gamma, which is itself cytostatic. And so if you think about this picture, the tumor cell actually has to participate in its own destruction. And so we know that tumor cells need two things. They have to be able to respond to interferon gamma and express MHC class 1. And the response to interferon gamma is very likely connected in that it's the thing that usually upregulates class 1 expression in the first place. And so without this MHC class 1, the tumor cells are pretty much invisible to CD8 T cell mediated recognition. And so if you think about this, there are actually many single gene defects that can lead to acquired resistance. And so this is a bit troubling, but if you think about it, there are multiple genes involved in interferon sensing. There are multiple genes involved in MHC class 1 antigen presentation, including, importantly, the light chain beta-2 microglobulin, without which you cannot get expression of any of the MHC class 1 alleles. There are, of course, non-genetic means of acquired resistance. And as a shout out to my colleague Judith Igudo, there are many non-genetic factors such as dormancy and hypoxia that can also lead to acquired resistance. But coming back to these genetic alterations, if we think about how are we going to overcome this so that we can re-engage the immune system, if the tumor loses the ability to recognize interferon gamma, you could possibly still induce class 1, maybe by induction of interferon alpha or beta, for example. But if you actually lose the class 1 pathway itself, it's very unclear how we get around this problem. And so, perhaps NK cells could come to the rescue, but this loss of class 1 is a central problem in cancer immunotherapy, particularly thinking about resistance and a lack of response in the first place. And so to study this in mice, my lab made models of primary and acquired resistance. We first used a very common cell line that responds well to checkpoint blockade, MC38. It's a colorectal cell line that has lots of mutated antigens. And I showed that it responds to PD-1 blockade, but if you delete beta-2 microglobulin, it cannot express MHC class 1, and we lose the response to PD-1 blockade. Pancreatic cancer is a model that my lab studies quite a bit. It does not respond to combination CTLA-4 or PD-1 blockade. So this is a model of primary resistance. And if you delete beta-2 microglobulin, of course, it still doesn't respond. What you will notice here, though, is that in teal, I'm showing you treatment with an agent we'll get to in a moment that we're very excited about, that actually seem to control and even induce long-term remissions in all of these models, including the ones that were beta-2M knockout. And so what are these IAP antagonists? And so these are small molecule peptide mimetics of SMAC. The structure of one of them is shown here, that operate downstream of TNF family receptor signaling. And so TNF receptor, as well as TNF family, superfamily receptors, are trimers. They associate in their cytoplasmic domain with TRAF2 and TRAF3, as well as these E3 ubiquitin ligases, CIP1 and 2. These small peptide mimetics can bind to the E3 ligase domain of CIP1 and 2, causing them to autoubiquitinate and degrade so that CIP1 and 2 protein are lost from cells. This allows the natural target of CIP1 and 2, NIC, to build up in cells and allow constitutive ligand-independent activation of the NF-kappa B2 pathway. So these drugs were originally developed trying to sensitize tumors to their own production of TNF-alpha. That doesn't seem to work particularly well across many tumors. But what we actually first reported, actually back when Glenn Dranoff was still here, and we've continued to study this, is that the IAP antagonists really enhance the function of many immune cells. And so I mentioned this TNF superfamily. Many of these members are expressed across the immune system. They tend to be activating receptors on immune cells. And so across the board, most things do better. So B cells become growth factor independent, dendritic cells produce more IL-12 and are better antigen presenting cells. CD4, CD8, NKT cells all proliferate more and make more cytokines, as do NK cells. And so we had shown that many of these cells in isolation are better activated, and that in the setting of a vaccine or in tumors that had a very robust T cell response, that you could induce anti-tumor immunity. And so looking on the T cell side, what we found is that the IAP antagonists don't activate all T cells equally. And so they enhance T cell proliferation, but they also skew their cytokine production. And so it's not that every cytokine is made more, but CD8 cells will make more IL-2 and interferon gamma, whereas CD4 cells, for example, will make a bit more of the Th2 cytokines. And so putting this all together, we wanted to look in a model of pancreatic cancer, one of these poorly immunogenic models. I showed you initially that it responds particularly well to these IAP antagonists, even in the absence of beta-2 microglobulin, which we'll come back to in a moment. But just to first show you that the cancer cells themselves are not affected by these drugs. These are isolated tumor cells in culture. We have two different pancreatic cancer cell lines, 6694C2 and 6499C4. And you can see by immunoblot that treatment with the IAP antagonist does induce loss of CIP1 protein. This is a degrader. It was invented before the term degrader was, but nevertheless, it self-degrades. And yet, the tumor cells themselves are perfectly happy and viable, even in the presence of supraphysiologic concentrations of TNF-alpha. So you cannot kill these cells with this drug. However, if you implant pancreatic tumor cells into mice and you treat the mice with IAP antagonists by oral gavage, you can see that the tumors here subcutaneously will grow progressively. But if you treat mice with the IAP antagonist in teal, these tumors will regress. Many of the mice in this setting are actually cured, and we can take these cured mice, re-challenge them with the same tumor in the absence of further therapy. They're protected from re-challenge, demonstrating immunologic memory. You can take the spleens from these re-challenged mice, stimulate them against two different, either an unrelated or this cognate tumor cell line, and show that there are tumor-specific T cells by interferon gamma ELISpot. And so this is very nice. This is actually a T cell high model of pancreatic cancer where we could demonstrate this very robust activation of tumor-specific T cells. However, most pancreatic cancer is not actually T cell high. And so coming back to this finding that IAP antagonism could be effective even in sort of these very checkpoint blockade refractory tumors and tumors that didn't even express class 1 on their surface, we wanted to explore this a little bit more deeply. So we next moved to an orthotopic setting. Pancreatic cancer does not grow in the flank of the mouse typically, so we surgically implant these tumors into the mouse pancreas. These are two different tumor cell lines, again, 6694C2 and 6499C4. And you can see in both cases that the orthotopic tumors are unaffected by PD-1 blockade. So if you compare the two gray bars, there's absolutely no difference. But if you look at the IEP antagonist in teal, you can see a consistent 50% reduction in tumor size, showing that even as a monotherapy in these checkpoint blockade resistant tumors, that the IEP antagonist outperforms PD-1 blockade. The same held true in a spontaneous mouse model. This is a KPC-GEM model where we have induction of oncogenic KRAS and loss of P53 in the pancreas. These are very aggressive tumors where the mice succumbed within 9 days of detection by ultrasound, but we could double this lifespan with IEP antagonists, and addition of checkpoint blockade did not really seem to change that outcome. Okay, so what is it doing? So we wanted to really explore the immunologic mechanism of action of what was happening in these tumors. Knowing that the IEP antagonist itself did not really affect the cancer cells, what was it doing to the microenvironment? And so what I'm showing you here is a panel of different genetically modified mice, where we have wild type mice here in black that show a nice 50% reduction in tumor size when treated with the IEP antagonist. But you lose this effect in mice that are lacking cross-presenting dendritic cells, CD8 T cells, alpha beta T cells. But CD4 cells and B cells seem to be not essential. And so from this, we concluded that it really was CD8 T cells and cross-presenting dendritic cells that were required for this immunotherapy in pancreatic cancer. We confirmed these findings with depleting antibodies. So this is looking at depletion of CD4 and CD8 T cells together. And again, you lose the therapeutic benefit of the IEP antagonist, really demonstrating that this is a T cell mediated therapy in pancreatic cancer. And so how could this possibly be? So we went back and looked again more closely at whether class 1 recognition on the tumor cells is actually important. And so just to verify that our beta-2M knockout cell lines do not actually express class 1, this is flow cytometry showing you on the top graph in black, pancreatic tumor cells really don't express very much class 1 at baseline, but if you treat them with interferon gamma in blue, you can dramatically upregulate class 1. However, you cannot get any class 1 expression in the beta-2M knockout tumor cells. They are still responsive to interferon gamma though, as demonstrated by upregulation of PD-L1. And so we checked not just beta-2 microglobulin, but a panel of different genes that are known to affect class 1 expression on tumor cells. And so if we looked in tumor cells now across the bottom that are deficient in beta-2 microglobulin, or TAP1, an important component of the antigen processing machinery, or interferon gamma receptor 1 or 2, you can see that across the board, the IEP antagonist is still able to reduce tumor burden, even when these tumor cells do not express MHC class 1. And so this was very puzzling to us. I had just shown you in the last slide that this was a CD8 T cell mediated therapy, and here we have tumor cells that are actually invisible to CD8 T cells. And so we thought, okay, well, maybe it used to be T cell dependent, but now we lose class 1 and it could have switched to an NK cell mediated mechanism. And so we tested that. We looked in beta-2 microglobulin knockout tumors and depleted NK cells. And lo and behold, NK cells are not required at all. But if we deplete T cells, you can see that this is still a T cell dependent therapy, even in beta-2M knockout tumors. We also used a genetic system to ask the same question, where we look in mice that are wild type mice. Again, very nice response to the beta-2M knockout tumors. In mice that lack perforin, a critical component of perforin granzyme mediated cytotoxic pathways. So this really knocks out the direct killing activity of both CD8 cells and NK cells. You still see that there is a very nice reduction in tumor burden. However, if we lack cross-presenting dendritic cells, you lose this effect again. And so altogether, these data really strongly indicate that direct recognition of MHC class 1 on the tumor cells is not required for T-cell mediated killing of the tumors. And so this is sort of the paradox we were left with, right? We have IP antagonists or an immunotherapy in pancreatic cancer. We know CD8 T cells are required, but they're not actually particularly abundant. I didn't show you these data, but the frequencies of CD8 out of total immune infiltrates can be, as low as 1% or less. That class 1 expression on the tumor cells is dispensable, as is the perforin granzyme pathway. And so this left us with two major questions. What do CD8 T cells do, and who actually kills the tumor cells? So we took a step back and we just said, okay, let's profile these tumors and see what immune cells are present. And so this is looking by a single cell transcriptional profiling at CD45 positive cells out of tumors from either vehicle or IP antagonist treated mice. And you can see that there are a large cluster or collection of clusters of granulocytes. There are several clusters of macrophages. Together, these form the majority of the immune infiltrates. And you have a teeny cluster of T cells, B cells, and a few dendritic cells. And so when we looked at what was different between the vehicle and the IP antagonist treated tumors, most things were the same, except for this one cluster of macrophages that I am tentatively calling macrophage phagocytic. And so what do I mean by that? So this really redirected our focus, and we said, okay, well, macrophages, what could they be doing? Usually in tumors, we think of them there for a wound healing, tumor promoting kind of role. But their primary job in the immune system is to fight off bacterial pathogens. And so, could it be that that's actually what they're doing in these treated pancreatic cancers, is trying to phagocytose not bacteria in this case, but actually tumor cells. And so this was really our first hint of this, where I showed you that if we looked at all the different clusters, the only one that was really increased and actually not present much in vehicle was this phagocytic cluster. And if we looked at the cluster defining genes, they were clearly macrophages, but they had genes that were very diagnostic of T cells or B cells. So the actual T cell receptor, B cell receptor, CD3 complex was showing up inside these macrophages. And at first I dismissed this and said, oh, they must be doublets. This is some kind of artifact, but they're not doublets. We looked very carefully at the RNA read counts and mitochondrial read counts to try to exclude this. And then we thought, maybe these actually are phagocytic macrophages that are phagocytosing lots of stuff, including tumor cells. But because T and B lymphocytes have high expression of truly unique genes, that it's a little bit of an artifact of the TINEC system, that this is what pops out as being phagocytosed. We did go back and add to our reference genome both KRAS and YFP and CRE to try to see if we could find evidence of tumor cells inside these macrophages. And we do see a little bit, although it's not as striking as the lymphocytes. So this gave us a hypothesis that perhaps what the IP antagonist was doing was actually increasing phagocytosis in vivo. And so, you can get a lot of data from these transcriptional studies, but at the end of the day, this was a hypothesis. We had to go look in vivo and actually see if this was what was happening. And so here is a picture by immunofluorescence staining of a tumor section where you can see macrophages in green that sort of look like they're engulfing these red tumor cells. And so we started to collect some of these images and think maybe this might be what's happening, but how could we quantify this on a larger scale? And so we turned to flow cytometry. We generated pancreatic tumor cell lines that had an M-cherry fluorophore, And so if you inoculate these cherry-labeled tumors into mice, and then you come back 2 weeks later and look at the tumor infiltrates, you can gate on just the macrophages shown here and ask which macrophages seem to have cherry labeled signal in them. And so in the boxes here are macrophages that we hypothesize have taken up bits of tumor debris, thus making them cherry positive. And so you can see that this is dramatically increased by the IAP antagonist, and this is just quantified over here on the right. So to convince you that these really were macrophages with internalized tumor fragments and not just two cells stuck together, we looked by imaging flow cytometry to see what was actually in that gate. And you can see that there are individual macrophages here, as indicated by CD11b staining, that have internal punctate structures that look like internalized tumor cell fragments. And so not an artifact of flow, these really are macrophages with internalized tumor material that are induced by these IAP antagonists. So given a proposed mechanism of action involving phagocytosis, we of course thought about the most famous, well, actually really one of the only receptor ligand interactions, CD47 on tumor cells and SIRP-alpha on macrophages that have been reported by Irv Weissman's lab to really be regulating this phagocytic process. And so CD47 is a don't eat me ligand that is expressed by normal healthy cells, particularly red blood cells, that prevents their phagocytosis by macrophages, and it can be upregulated by cancer cells. And so what we did is we first took tumor cells and we deleted CD47 from the tumor cells. You can see that this already makes them grow smaller, suggesting that there's some endogenous level of macrophage clearance or control of these tumors that can happen in the absence of CD47. And if we treat these tumors with IAP antagonists, you can see that they respond quite nicely. Genetic knockout is not a therapy, so we also, in collaboration with Chris Garcia and Hida Plu's lab, had helped develop a CD47 nanobody from alpacas, and I can show that if you treat mice with this CD47 nanobody, that you can again sort of at least combine with the IAP antagonist for reduced tumor growth. Okay. And if we take this CD47 nanobody and we put it into the genetic mouse model of pancreatic cancer, again, this KPC-GEM mouse, you can see that we were able to extend survival beyond just the IAP antagonist monotherapy, which is better than we had been able to do with checkpoint blockade. So again, suggesting that agents that sort of further increase phagocytosis could be combined with this IAP antagonism. So we got off on a tangent. Does it actually require T cells? I mean, I started by telling you that this was a CD8 T cell mediated process. So is there anything about this phagocytosis that requires a T cell response? And so to test this, we again looked at cherry labeled tumor cells and this in vivo phagocytosis by these macrophages and asked, is the phagocytic process affected by T cells? And so you can see again this increase in phagocytosis by the IAP antagonist in wild type mice, but in BATF3 knockout mice that cannot make antigen specific T cell responses because they lack cross-presenting dendritic cells, you see that this response is greatly blunted. And so something about these phagocytic macrophages still required an antigen specific response. So this led us to the question, what are the T cell derived factors that influence phagocytosis? And we made two assumptions. We assumed that first, it was probably a secreted factor. We have a small number of T cells and a large degree of space and lots of macrophages. So a soluble factor would be more efficient at inducing or at contacting each of the macrophages. And we hypothesized that it had to be increased by IAP antagonism. And so to get at what this might be, we first looked at CD8 T cells activated in vitro, and we asked sort of which cytokines were increased by IP antagonism. And you can see a panel of options here. We asked then, of these cytokines, which ones were actually being produced in pancreatic cancer? And just because a T cell can make it in a dish does not mean that it makes it in the tumor. And so we took orthotopic tumor infiltrates from our pancreatic cancer. There are not very many T cells, but we could pool enough tumors to sort the few that were there and perform limited bulk RNA-seq. And we showed that of these candidate cytokines, the two that were most expressed were lymphotoxin and interferon gamma. But if we looked at which were actually increased, it was really just these top three that were significantly increased by IMP antagonism. And so to really test, which of these candidates was actually responsible for the immunotherapy benefit of IMP antagonism, we used both genetic loss of interferon gamma and also blockade of these different cytokines with blocking antibodies, or in the case of lymphotoxin, a dominant negative version of the receptor that has an Fc. And so just to walk you through this, these again are poorly immunogenic pancreatic tumors, where in wild type mice, we get a 50% reduction in tumor size. This is still significant in interferon gamma knockout mice, but not if you treat those interferon gamma knockout mice with a blockade of lymphotoxin. Likewise, if we have all wild type mice, but we treat with blocking antibodies to TNF alpha, absolutely no effect. So this is still quite significant. But if we block lymphotoxin, we lose the efficacy of IIP antagonism, as well as if you block lymphotoxin in combination with interferon gamma. And so this really pointed us to lymphotoxin as being the T cell derived factor that influenced phagocytosis and the reduction in tumor burden. So we wanted to know a little bit more about what lymphotoxin was doing. This is sometimes difficult to interpret at this high level view where you're looking at tumor size as a readout. And so we wanted to get a more in vitro reductionist system where we could really ask, how are these macrophages actually being activated to phagocytose tumor cells? And so we set up a reductionist system in vitro where we used bone marrow derived macrophages and still are cherry labeled tumors where we could try to co-culture cherry tumor cells with macrophages and ask again by flow cytometry what the rate of phagocytosis was. And so if you just put macrophages and tumor cells together, absolutely nothing happens. If you add lymphotoxin, lymphotoxin alone doesn't do anything either. But if you add the IIP antagonist, you can see that there is a little bit of phagocytosis occurring, and that if you add IIP antagonist with lymphotoxin, this is dramatically increased. The effect really seemed to be on the side of the macrophages. If we pre-treated the macrophages with IIP antagonist and lymphotoxin, you could get this increased phagocytosis. But if we pre-treated just the tumor cells with the IIP antagonist or lymphotoxin, really nothing happened. And so this looked like it was a combination of this altered NF-kappa B signaling in macrophages combined with this exposure to lymphotoxin that really changed the macrophage from its baseline phenotype to being this tumoricidal macrophage that could really engage in phagocytosis of these tumor cells. So this is not typically how we think of macrophages. Macrophages in their regular day jobs, they maintain homeostasis by clearance of dead and dying cells. And so do they really phagocytose live tumor cells? So this is just a review from Doug Green's lab looking at what we know about macrophage-mediated interactions with both dead and live cells. And so they can typically recognize apoptotic cells by exposure of phosphatidylserine on the membrane of the apoptotic cell or binding of calreticulin. And so they're quite good at recognizing dead cells and inducing phagocytosis. But live cells have some don't eat me signals the best understood is, of course, CD47 that I already mentioned. And then there are a few others that have only been shown in humans to block phagocytosis. And so this is just a video to kind of illustrate what that cartoon showed, in that we have macrophages here in gray, and we have tumor cells in yellow. There is also a blue dead cell marker in the media. And so you will see that these macrophages are going to crawl all around the plate. They're going to have plenty of opportunity to eat the yellow cells, but they don't. But now a cell died in the middle, and they just gobble up the dead cell, right? And so this is what macrophages do. They are there to survey for apoptotic bodies and clear out the dead remnants, but they do not touch the live tumor cells. And so to really test whether we were getting live cell phagocytosis, we wanted to see this in action to really convince ourselves. And so here is the same setup where, again, you have macrophages in gray, tumor cells in yellow, but we have pre-treated these macrophages with the IP antagonists and T-cell derived cytokines. And so now what you will see is the macrophages are engaging with the live tumor cells, and it grabs it and pulls up and eats the live tumor cell. And if you look fast, there are 5 of these that pop up around the screen, where the macrophage is really interested in that live cell, engulfs the live cell, and you don't see any blip of dead cell blue marker until it is already inside the phagolysosome. And so this is the only blue you're going to see is the remnant of the tumor cell that had been eaten. And so this was super exciting. I really want to thank Dr. Lee Kang, who is the fellow in my lab, who really put a lot of effort into getting these beautiful movies. They are 18 hours of video microscopy that were going on through the night. And so we thought that perhaps we needed a more high throughput way to quantify this live cell phagocytosis. But this is really a difficult problem because all of the static readouts that we have are very hard to distinguish whether you're phagocytosing live versus dead cells. And so, I mean, I've showed you flow cytometry, immunofluorescence, even imaging flow cytometry. How do we know that these macrophages engulfed a live cell versus the tumor cell died by some other means and the macrophage engulfs the remnants, and that's why it looks positive. And so we thought about this for a long time and realized that only live cells have intact nuclei. And so if you're looking here, this is a confocal picture of a macrophage in red. It has its own nucleus in blue, but you can see it's also engulfed this green tumor cell, and that the tumor cell has its own nucleus. And so although we can't always determine whether the tumor was live or dead, this tumor had to be alive when it was eaten by this macrophage. And so Lee looked through lots of images, kind of using these criteria as to whether we have some punctate structures indicating sort of total phagocytosis versus the rare cells we could catch in the act of having just engulfed a live tumor cell that still have intact nuclei. And so if you measure across many images, tumors there are cells that were treated with IP antagonists versus vehicle, you can see that in the case of vehicle, there's really almost no phagocytosis, but that you could really induce a strong phagocytic phenotype with the IP antagonists. And it was in those IP antagonist treated cultures were the only times that we ever saw these live cell phagocytic events with intact nuclei. This dramatically underestimates the frequency, but at least in these cases, we can say that it had to be a live cell at the time it was engulfed. And so this has led us to a new model of thinking about how we can use immunotherapy for some of these recalcitrant tumor types. And so what I've shown you is that we have an agent, these IP antagonists that induce non-canonical NF-kappa B signaling. They actually do this pretty pleiotropically across many immune cell types. And so we know that this increases the efficacy of dendritic cells, but importantly, it polarizes the T cells to secrete lymphotoxin that can then act on the macrophages to generate these tumoricidal macrophages. these macrophages can then go and engulf and kill the live tumor cells. And it really doesn't matter if these tumor cells express class 1 or not. And so this is a potential way of actually retaining a degree of antigen specificity for what would otherwise be a checkpoint blockade resistant tumor. And so this is very exciting. It leaves us with two very important questions. One, does this happen in people? All the data I've shown you has been in mice thus far. And two, what is regulating this process? How does a macrophage actually recognize a live tumor cell? And how does the tumor cell then get eaten by the macrophage? And so to address the first question, we realized that we could use our in vitro system to model what might be happening in humans. And so we tried looking at human macrophages from peripheral blood monocytes and co-cultured them with human pancreatic cancer cell line. And what you can see is that the human macrophages, when exposed to IP antagonists and human lymphotoxin, do readily engulf and phagocytose these human tumor cells. And so we've done this now across a panel of human pancreatic cancer cell lines. We're actually very excited about using these to screen for factors in an all-human system that might affect this process. And so thinking about what makes the tumoricidal macrophage, we were really curious about this. And so we tried a transcriptional profiling-based approach where we took macrophages from pancreatic tumors ex vivo, as well as the ones I've shown you in vitro culture. And although there are a lot of differences between in vivo and in vitro, the common feature between these two systems is the ability to phagocytose tumor cells. And so we looked at these 473 genes that were in common. About 90 of them are upregulated surface proteins on the macrophages. And so we are very excited about deleting these using a CRISPR-based technology and figuring out which ones are actually important for the macrophage uptake of live tumor cells. On the tumor cell side, we also wondered what factors the tumor cell had to express to be taken up by these tumoricidal macrophages. And so we did an in vivo CRISPR screen that I'm showing you here, sort of modeled off of what was originally set up in Nick Koenig's lab when he was still here at the Farber. And we have mice that were treated with either IP antagonists or with combination checkpoint blockade. And so the IP antagonist tumors are quite small. This allowed for us to have a Darwinian selective pressure. And when we looked at the genes that were potentially conferring resistance, we found a convergence on the TGF-beta pathway. And so this is something that we're definitely following up. There's obviously a large literature on TGF-beta in pancreatic cancer. And so whether this is related to particular ligands or the EMT in general is something that we are actively investigating. But this really brings up an important point that this field of macrophage tumor cell interactions, which has thus far really just been dominated by CD47, is the tip of the iceberg. We think that there are lots of these receptor ligand interactions that are important. And just to give you a piece of evidence here, if we look at our tumor cells that are phagocytosed nicely by macrophages that have been termed into tumoricidal macrophages, if we do the same thing with CD47 knockout tumor cells, you can see that there's still a dramatic increase. And so whatever the tumoricidal macrophages are doing, it's not redundant to CD47. There is some orthogonal pathway, either new activating receptor ligands that are induced or inhibitory receptor ligand interactions that are diminished. But it's not just CD47. And so to really find these in a high throughput fashion, we've developed an in vitro CRISPR screen where we take macrophages, co-culture them with tumor cells, and sort back out both the tumor cell fraction and the macrophage fraction. We've done enough pilot tests to assure ourselves that not all of the DNA is degraded immediately in the macrophage, and so we can recover enough of these tumor cell DNA from the macrophage phagolysosome to be able to perform a CRISPR screen. So I'm unfortunately going to leave you hanging on that one, but this brings me back to this question of, how common is this process? And so throughout this project, we started wondering, do CD8 T cells usually kill tumor cells directly, or is this kind of indirect mechanism more common than we might have appreciated? And, we work on a lot of different immunotherapies in the lab. And so we started to test our beta-2M knockout tumor cells across multiple different immunotherapy modalities, and sort of reassuringly check our PD-1 blockade and IL-2 are both requiring CD8 T cells and requiring class 1 of the tumor cells, as we would predict. But many of the other pathways we're looking at do not actually require MHC class 1 on the tumor cells, some of which, however, are still CD8 dependent. And so I think this is actually more common than we might have anticipated. And we started to look into a lot of these other mechanisms of sort of T cell indirect means of killing tumor cells. With that, I will thank the many people in my lab who contributed to this over the years, particularly Kevin, who was the main driver of the project I just showed you, as well as Lee, who is doing all of the work on the macrophage screens that I talked about at the end. A huge shout out to the Dana Farber Hale Center. This is a really fantastic group of people who are all focused on pancreatic cancer, and we have really excellent collaborations with all of them, thinking about how to bring our various expertise to bear on this very difficult disease. Much of this research was actually sponsored by Novartis that makes the IAP antagonist that we were studying, although many companies have similar compounds. So with that, I will also show the Hale Center group and put my model up at the end and take some questions.
Thank you so much. That was really a remarkable presentation. So there are a lot of questions. I'm going to start by Patrick. I apologize in advance if I mispronounce somebody's name. I will try my best. I do know that it matters. I like it when people pronounce my name right, so I will try my best. So Patrick Anderson is asking, excellent talk. Do you think that T cells also have a role downstream of macrophages following antigen presentation of the engulfed live tumor cell?
This is a good question. I do think naive T cells have to be primed by a dendritic cell that both preserves the antigen long enough to present it on class 1 and class 2, as well as providing co-stimulation. Now, whether the T cells who are already effector cells and in the tumor could engage with the macrophage in this kind of setting, I don't actually know. So that is a very good question. Clearly, the class 1 is being provided by something. We think that it's the cross-presenting dendritic cell, but it, of course, could be any host cell that still retains class 1.
Takashi is asking whether KRAS or P53 mutations are required to see the effect that you reported.
That is a good question. We don't know. We've mostly looked in pancreatic cancer models that, of course, all have KRAS mutations. As we're doing these CRISPR screens on the tumor cell side, I think it will become apparent what the tumor cell actually needs to express. And is this something that any tumor could do or be sensitive to, or is this something that, a particular genetic subset would be more amenable? And so this is something we're actively looking into.
Oluwakemi is asking, thank you for this beautiful presentation and interesting videos. Can you give a rough estimate of how long it will take for a tumoricidal macrophage to clear live tumor cells in a mouse?
Well, it's, with any immunotherapy, there's always a race between sort of the immune system clearing the tumor and the tumor proliferating. And so in some models, the subcutaneous ones I showed you, we could actually cure mice. So the macrophages win in the end. But in some models, the tumor is still slowly growing out. If you're asking about, the video is 18 hours long. So how long does does an individual macrophage take in vivo? I don't actually know. It probably still is on the order of, an hour or so as it's crawling around. We could probably try to estimate from the time in the video how long it would actually be for full tumor digestion to happen. It's a good question.
Takashi again is saying, I think you mentioned that macrophages ate live cancer cells. However, in dying cells, phosphatidylserine exposure is started before their death. So I would like to know whether LCL treatment increases phosphatidylserine exposure in cancer cells when they are still alive. In addition, I would like to know whether lymphotoxin treatment increases phosphatidylserine receptor expression in macrophages.
Great. Yes. Thank you for that skepticism. We do think this is really important to show that the tumor cell is either alive or dead, or where it is in that process when the macrophage first contacts it. What I can say is that in vitro, there is absolutely no cell death induced, no phosphatidylserine expression, nothing. We've even done full transcriptional profiling on tumor cells exposed to these IP antagonists, and there's like nothing that's exchanged by twofold. So in vivo, it's a little bit harder of a question to answer, but we don't see that the phosphatidylserine receptors on macrophages increase. So whatever's making the macrophage turn into this tumor cell macrophage is not just an upregulation of GAS6 and some of the other known receptors. So yeah, I think it is a very difficult question to really prove a negative there, but at least it doesn't seem to be the main mechanism in our opinion.
Edward Fritz is asking, he's saying superb seminar. Do these tumoricidal macrophages present antigen and lead to new specificities? How do you explain adaptive memory?
Well, the tumors have antigens. And so, they had antigens going in, they still have antigens coming out. What these are, we didn't actually identify. We could find tumor-specific cells by ELISpot, but that doesn't tell you, what antigen it actually is. It's probably not a neoantigen. If you sequence these tumors, they don't actually have any protein coding changes that are predicted to encode for neoantigens. But there are lots of self-antigens. I mean, there are many ways that tumors can be seen by the immune system that is not just mutations in protein coding sequences. The antigens recognized are not likely to change during the course of this treatment. If you do have new antigens being revealed and they're not clonal, then it's not likely to be something that would be very important in the T cell response. So, yeah.
Yinghua is asking, if you treat the macrophage tumor co-culture with CD47 blocking antibody, do you observe increased phagocytosis to live tumor cells compared to LCL treatment?
We have looked at this. And so in vitro, we can recapitulate that CD47 nanobody will increase phagocytosis. It seems to be both the live and the dead cells. So we've started including a dead cell marker in our in vitro assays to try to distinguish that. But what I can say is that you can make more phagocytosis with IAP antagonists. And so while CD47 seems to be potentially part of the pathway, it's not the only receptor that's important on tumor cells.
Juan Osorio, this one I can pronounce right. Have you tried other tumor cell lines as target in this system? Do you think size of the tumor cell influences their capacity to phagocytose live cells?
Yeah, so you would be impressed by what a macrophage can actually eat. We have not found a tumor cell that's like too big for the macrophage to, move its membrane around and phagocytose it. And so they eat things that are their same size or even bigger. Maybe some of these giant cell tumors would be difficult, but I'm not. We don't have them cultured particularly well to test that. What was the first part of the question again?
Yeah, if you have tried different cell lines and
Yeah.
whether the size had.
Right. So we've mostly looked at a panel of pancreatic cancer cell lines. So we've looked at several mouse ones and several human ones now. So we are expanding this to more tumor types.
Kandan Slay, awesome presentation. Thank you. Have you looked into macrophages presenting tumor antigens to Th cells after engulfing live tumor cells?
So again, I'm not sure that a macrophage can really activate a naive CD4 T cell. Whether it could provide class 2 presented antigens that would help, further activate effector CD4 cells in the microenvironment, that certainly is possible. So whether that's important here, I don't know. We did look in class 2 deficient mice, and you can still get very nice tumor reduction when treating with the IEP antagonist. So, that does tell you that, at least in this case, neither CD4 cells nor class 2 are critical, although, could they be playing, an auxiliary role potentially. CD4 cells do make lymphotoxin. So I don't want to completely rule them out as a contributing factor.
Can you apply these to chemotherapy-induced efferocytosis?
Yes. So, macrophages do readily take up dying cells. And so, efferocytosis has a name because that's what macrophages mostly do. We can show that, certain things that are known to regulate uptake of dead cells, like, for example, calreticulin, we can really see that if you knock that out of macrophages, that they are, less able to take up dead tumor cells. But the IEP antagonist really does enhance live cell uptake. I guess your question of whether, if we killed all the cells first with chemotherapy, would they be better taken up by these macrophages? I don't know, because normal macrophages are so good at it that the baseline's already, 100%.
Is the LCL treatment toxic? Have you tested if the tumoricidal macrophages can engulf normal cells?
That is an excellent question. The model here at least has a degree of antigen specificity in that the macrophages are only activated in the vicinity of T cells that are tumor specific. And so, it's not that we look in the mice and like every macrophage is activated. You don't have that kind of phenotype. And so there's some spatial restriction provided by the need for T cells. In humans, the IEP antagonists have gone through phase 2 clinical trials. And so both the Novartis drug and several of the other ones, notably there was a successful trial by W Farm in head and neck cancer combined with radiation. And so the toxicities that have been seen in people dosed with these compounds are restricted to transient upregulation of cytokines. And so you get, a small cytokine burst at, 24, 48 hours that goes away. And that's kind of consistent with what I told you in the beginning about these agents activating cytokine production from T cells. But you don't really see a lot of macrophage damage across the different organs. So I think that they are fairly well tolerated. So I think that there's a path forward.
So I do have myself a question regarding this engulfing of normal cells, and you very nicely proved that class 1 is not necessary, but is loss of class 1 actually helping and improving this eating of the tumor cells, or it's the same regardless of class 1 expression?
Yeah. So we did directly compare the rate of phagocytosis between class 1 positive and class 1 negative tumors, and we didn't see any difference. And so I don't think that loss of class 1 made them better phagocytosed. We've not looked at a lot of cultures of normal cells. I think that is actually quite important. And really the only thing we've done is looked at co-cultured fibroblasts that sort of accidentally came with one of our ex vivo cell lines. But this is something that we can actually readily address by looking at, for example, co-cultured spleen cells. Normal spleen cells, are they actually taken up? So, we did find them by transcriptional profiling that identified T and B cell genes in there. So I don't want to tell you that they will never eat a normal cell. But yes, we do need to see sort of what the therapeutic index is between cancer cells and normal cells.
Okay, so it's already 1. There are a few questions. Maybe I can read the last one. I mean, there are still some questions that I didn't read. So if you still have questions and I didn't read them, please type them in. We will send them to Dr. Dugan and she can answer to you. So don't be afraid that the time is running out. Amazing talk from Juan Rodriguez Vita. Amazing talk. Is it possible that IAPs inhibit also the don't eat me signal receptors? Like they control CD47 expression?
So it's not via CD47. There certainly could be some don't eat me receptors that are, reduced by the IP antagonists, but this is exactly what we're looking for. So the CRISPR screens we set up would find either positive or negative regulators.
Perfect, thank you. So we will send the remaining questions to Dr. Dugan. Dr. Dugan, thank you so much for such an interesting presentation, teaching us a lot about what macrophages can do that we were not thinking about. And yeah, thank you everybody for attending the seminar today. Thank you.